Okay, for virtual microscopy, we are going to start with the appendix. Okay, so just to orientate you in the beginning, so an appendix is a tubular organ. So there is a lumen in the center, and it is lined by a layer of mucosa and an outer muscular wall, and there is also attached fat, which we call mesoappendix. Okay, so in inflammation of the appendix will first involve the mucosa, and it will also involve the wall of the appendix. So look at these tubular structures in here. So these tubules or glands, these are your mucosa lining, and here you can see them, which means they are intact, and then there are areas which they have been lost. So the area of loss is what we call ulceration. So remember, that is also a manifestation of acute inflammation. Okay, so now we zoom in. All right, so some of the areas, and let's hunt for neutrophils. So neutrophils are the key cells involved in the process of acute inflammation. So how do you recognize them? Neutrophils are cells that have multilobation. So they look like a Mickey Mouse, essentially. So you can see a head and two ears, head, two ears, head, two ears. Okay, so if you move along and around the appendix, you will see there's actually quite a few of them scattered about. Okay, so head, two ears. So all these Mickey Mouse structures, these are your neutrophils. Okay, so that is the first thing you need to identify. Okay, and they have spilled all the way into the muscular layer and into the fat. Okay, so next you want to look for the vascular congestions. Okay, so vessel dilatation and congestion. Usually the vessels are not that obvious. So in an inflammation, there will be an increase in diameter, there'll be more blood flow, so they will look more red and congested. So these are all your dilated and congested vascular channels. Okay, so see, these are all very open and filled with red blood cells because these are now congested and so full of red blood cells. Okay, and then the other thing you want to identify is edema. So edema is fluid which comes out of the vessels into the interstitial space. So you don't see the fluid inside the vessel, but you see them outside the vessel. So that's clue number one. Clue number two, because fluid is generally clear, so they impart sort of a whitish or a pale sort of appearance to them. So this is an example of an area with edema. So these whitish sort of pale areas, this is all filled with fluid. And what it does is it pushes the cells and the structures far away. So you can see the cells are now pushed apart as opposed to areas where they are clumped together. All right, so in this region you can see dilated blood vessels and edema. So see if you can find the same structures elsewhere or the same changes elsewhere in this slide. So next I'm going to move on to the abscess formation. Okay, so remember that you can always use the small image here to guide where you are at. So right now I'm at this region, so you can see a very dark blue area, and this dark blue area is composed of, again, see, multiple Mickey Mouse structures. So this is neutrophilic infiltrate forming pus. So this is the abscess. Okay, so this is the content of the abscess, and an abscess is usually surrounded by granulation tissue. So granulation tissue is angiogenesis, so you see a lot of blood vessels, so small blood vessel formation. So we'll see that in the last case. Okay, right, so that's one piece. And if you move on to the other piece over here, again you see this very dark colored area, and if you zoom in you will see more neutrophil exudate. You will also see cellular debris, so these are dead cells because you can see the outline of the cells but no nuclei. Okay, and there's also some pinkish material in and around the region. Some of them may represent fibrin. Okay, so remember, neutrophilic exudate rich in exudate is rich in cells, rich in neutrophils, rich in plasma protein, and also cellular debris. Okay, and then granuloma. Okay, so granuloma, so on low power you immediately see areas of kind of light pink surrounded by really dark blue region, and this is a lymph node, so you can see a capsule all around it. Okay, So in caseating granuloma, which is what this is, there are four main components that you need to be able to identify. Okay, so remember that there is a central caseating necrosis. So necrosis means cell death. So you can no longer see the nuclei because the cells are dead, but you can see some granular material, you can see some cellular outlines remaining. Okay, so let's call that the central part. So in a caseating granuloma, the central necrosis is rimmed by epithelioid or activated histiocytes. So which means you will find the histiocytes immediately adjacent to the necrotic center. So if this is your necrotic center, then this is the region where you should find the histiocytes. So histiocytes are cells with elongated nuclei, which have been likened to footsteps in the sand. So if you walk along the beach, you can see your remaining footprints or slipper prints, and they look a little bit like this. So they're a bit elongated. Okay, so these elongated cells with eosinophilic cytoplasm, these are your epithelioid histiocytes. Okay, so all around here remain the necrosis. Okay, third component, your multinucleated giant cells. Okay, so these are generally easy to identify because they're really large. And remember that multinucleated giant cells are formed from fusion of multiple histiocytes. So you should find them in the region where the histiocytes are found. So normally in the areas remain the necrosis. So I'll just bring you to another place. Okay, just illustrate that point. So here you have necrosis. So this region is the area remain the necrosis. So that's where you find the histiocytes. And here you can find a giant cell. Okay, so there's a few more of those in and around the region. And then lastly, and the outermost region of a granuloma, you will find rim of lymphocytes. Okay, so these are lymphocytes. So lymphocytes are cells which have only one nucleus. So it is a mononuclear cell, and they have very little cytoplasm. So they just look like blue, basically, blue cells. Okay, so that's your granuloma. And lastly, this is granulation tissue. So granulation tissue by definition is new blood vessel formation. So what you should see is multiple and numerous blood vessels. So these are, okay, so I'm looking at the most right-sided piece here. You can see multiple blood vessels. Okay, and how do you tell they're blood vessels? They have a wall and they have a central lumen, and the lumen contains red blood cells. Okay, so multiple red blood cells. Multiple vascular channels. Okay, so these are all your granulation tissue, and granulation tissue often seen as part of healing, and there might be coexisting inflammation as well at the same time. And in this case, in between the blood vessels, you may be able to see all these inflammatory cells. Okay, on top of finding inflammatory cells, you will also find another population called fibroblasts. Okay, so fibroblasts are cells that are responsible to lay down collagen, and they are a bit more elongated in their appearance. Okay, so these are them, cells which have a lot of eosinophilic cytoplasm with tapering ends. Okay, right, so that's fibroblasts. Okay, so that's all fibroblasts. And the cells with the smaller nuclei and hardly any cytoplasm, that is your lymphocyte. It's not very obvious in this particular piece, so I'll just move on to another area. Okay, so here some of these kind of small cells with not a lot of cytoplasm, these are your lymphocytes. And of course, in between that, you will also see some other inflammatory component. You can see eosinophils, you can see some neutrophils, you can see macrophages, plasma cells. Okay, so these can all be part of a granulation tissue.